to share some of the science and the latest developments that have been happening in the HERN. And we're fortunate enough to be able to share a lot of the exciting developments that have been happening. Before we begin, I wanted to remind you that this meeting is going to be recorded and posted on YouTube. This is a public webinar, and a link will be sent to everyone afterwards. So please make sure to check yourself and make sure that you're muted. You'll have an opportunity at the end of the webinar to ask our presenter questions. Next slide. So what we'll do today is, for those of you that may not know about the HERN, I'll give you a brief introduction to what that is. And then we'll jump right into today's webinar. Next slide, which will give you an overview of some ways in which you can use islets for high density applications, and then show you how to use islets in whole culture, and kind of review some of the pluses and minuses of each, and show you how you might use some of this technology in your own laboratory. Next slide. So HERN is a fairly large network made up of several different consortiums, and you see each of these consortiums, the icons for each of these consortiums listed here. Each of these groups have a different theme that they focus on. The overall goal is to understand how type 1 diabetes develops, what ways we can develop strategies to try to prevent or protect human islets. And today we're going to be highlighting work from one of those consortiums, which is called CTAR, the Consortium on Targeting and Regeneration. Now, CTAR is investigating methods to increase or maintain functional beta cell mass in type 1 diabetes, and you're gonna hear a lot of the exciting science that's coming out of that group today. So next slide. In all, what we have is we have about 200 investigators participating in this network. There's a lot of different institutions and 9 different countries. Next slide. Now, in addition to all of this work, we do catalog all of these developments, and you can go to our website and you can download these reports. These are semi-annual reports that come out and that share a lot of the science. In addition to that, all of the publications that are coming out of the network are also found there. Next slide. In addition to the publications and the reports, next slide, there's also a resource catalog in which all of the data sets, the mouse strains, the cell lines, just the various resources that are being generated by this network are also being put in one place so that you can easily find that, have access to it, and contact the investigators who, in many cases, created these new networks. Next slide. And you can find that on the homepage of the HERN network. And when you go there, next slide, you'll see that there is a catalog in which you can click by different buckets. So you can click on bioreagents or data sets or different documents. Today, we're really going to be focusing on one of the protocols that Dr. Wagner has deposited there so that you can get access to that following this presentation or even now during the presentation. So with that said, before I hand it off to Dr. Wagner, I just wanted to give a brief introduction to our speaker today. Dr. Wagner earned her undergraduate degree in biological sciences from Harvard and then went on to get her PhD degree in biochemistry, also from Harvard. Following that, she did her postdoc at the Broad Institute, where now she is the director of the pancreatic cell biology, metabolic disease, and chemical biology program. Dr. Wagner has received several different awards and honors, which we won't cover today, but what I will point out is she is highly published, and in regards to today, back in late 2020, she and colleagues published a paper on a 3D culture platform, and she's continued to publish on that in 2021. And this is makes up sort of the basis of what we're going to be hearing about today. So without further ado, Dr. Wagner, thank you for taking the time to share your work and that of your colleagues.
Great, thanks, John. Oh, then there's the resource browser. Okay, so today I'm going to tell you about our efforts to culture human islets and use them for small molecule discovery. Again, with the main goal here is to make this a very accessible process for you. I hear a lot of people that come to us that want advice on that, and that's great. But it's my opinion that all of these techniques are highly doable. And actually, by taking a very systematic approach to this culture system, we think we can get around a lot of the donor-donor variability. We actually can see quite a lot of reproducibility across donors, which might be surprising. So to begin, my group, again, as mentioned, really takes a high throughput approach to small molecule discovery. And here we take phenotypic approaches to screening, in which case we're looking for small molecules that have an effect directly on the cell without regard to the mechanism that compound takes. Today, I'm going to focus primarily on beta cell proliferation, but the system that I'm going to describe can also work to look at beta cell survival, insulin secretion induced by glucose. And even some of the other work we've been doing is looking at transdifferentiation from one cell type to another and whether that can be induced by small molecules. But to do this, we started this over 10 years ago with the idea that, well, we'll take islets, we'll throw them in a dish, and we'll treat with compounds and then read out beta cell proliferation, because that's what we had done with cancer cell lines in the past. So why not do this with islets? Of course, never that easy. Human islets do not adapt well at all to plastic dishes. And so we needed to take a step back and look through the literature to understand, well, what's been done. And what we found is that over 20 years ago, actually, Alberto Hayek's group at UCSD had done this kind of analysis and comparison of the different substrate types for culturing human islets. And what they found was that this cell line, HTB9, is a human bladder carcinoma cell line. And the extracellular matrix that it secretes onto the plastic dishes is ideal for attachment of human islet cells for preservation of glucose responsiveness and insulin secretion. They had done this technique in larger wells where they're looking at, for example, even Petri dishes. So we decided, well, maybe we can adapt this to a 96 or 384 well culture system. And the way it works is that the ECM is secreted by the cells in culture as a kind of carpet that's between the cells and the plastic. And when we began this process, what we did was we grew these HTB9 cells to confluence, waited 3 days, maybe even longer sometimes, and then used ammonium hydroxide to decellularize the layer. And what that did was it takes the cells off, but it keeps the ECM intact. And we can stain the plate that no longer has cells for things like fibronectin and laminin. This is a electron micrograph showing that actually the surface of the plate, you wouldn't even be able to tell by eye the difference, but you can see it. It has quite a grainy surface, and that is the ECM itself. However, when we do this, we can then dissociate islets, we'll get to that in a moment, and culture them on top of this ECM surface. When we do that, we can also then, at the end of the system, the culture, stain them for insulin and glucagon, for example. And as we would expect from a human islet, we see a roughly equal distribution of alpha cells and beta cells in culture. But this decellularization process is really laborious, and especially if we're thinking about 384 well plates, it took a lot of automation and a lot of consumables. It became kind of a mess when we were starting to think, well, long term, if we wanted to really do a real high throughput screen, this process was not the best. And so we decided to take a step back again and say, well, can we even simplify the process and just look at the conditioned media secreted by the HTV9 cells? And so we do exactly that. We culture them in T75, T175 plates, wait a few days until they reach confluence. Then we wait 3 days after this. And all of these steps, again, are going to be in the protocol that I'll talk about at the very end that you can access online. So you don't have to take notes today. I'll talk about it, but it's all documented in the Word document. After 3 days, we collect the supernatant, and we can use that to coat the plates at 37 degrees. So really, it's quite easy. We just add, in the case of 384 well plates, 10 microliters per well. Wait a couple of hours, remove and wash once, and then they're ready to go. Now, when we did this, we could then also stain again for fibronectin, collagen, laminin, and we see, and sorry, these are not the best images, but it was kind of quite dim. It's hard to get a good image of these wells that are really, frankly, empty. But you can see that in an empty well, there's no staining. With just media, there's no staining. With our conditioned media, you can see kind of a granular surface, and that's exactly what we're looking for. And it's really remarkable. When we culture islet cells on this surface, it's almost, they're not going to fall off. It's almost difficult to think about ways to get them to even come off the plates, which could be a disadvantage, and we'll talk about that at the end. We could then compare the plate surfaces. For example, this is with the conditioned media versus the ECM cell-based method. And we plated some islet cells and stained with PDX1 in one case and C-peptide in the other. In both cases, the cells look pretty healthy and happy. And so we were pretty much content to move forward with this process. Again, when we do this and we compared then again the supernatant or the conditioned media versus the ECM and the collagen 4 coated plate that we could commercially purchase.
115.
It is 11.
Oh, hey, if everyone could mute, thanks. And we can see the total number of cells per well is pretty stable across the different surfaces. And the number of PDX1 positive cells per well, again, just highlighting the number of beta cells, we tend to get on the order of 30 to 50% beta cells in our culture system. Also, and very importantly, we can preserve beta cell function over time. And this is comparing the conditioned media with the ECM. I'm not even showing here uncoated because frankly, when we put them, the cells in to uncoated plates, they just don't stick at all. And, we get a very much overgrowth of the fibroblasts that are in the rest of the culture system. So comparing the conditioned media with the ECM, we get a really nice about 4-fold increase in insulin secretion at low and high glucose. We can culture these cells also up to, I think we've gotten to 2 weeks so far. We don't really need more than that, but I imagine you could test the limits and see if you could keep them growing even longer. I think really the limit becomes the overgrowth of non-endocrine cell types in the culture. Now, to measure proliferation, we use EDU incorporation. Quick review for those of you who are less familiar. EDU is ethinyl deoxyuridine, and it looks very, very similar to bromodeoxyuridine, which has been used for a very long time to look at incorporation and S phase of the cell cycle. However, the BRDU detection requires antibodies, and really it's inaccessible. The antibodies are inaccessible without denaturing, and so you need to kind of acidify histones. It really disrupts the cytoplasmic structure. You can get the nuclei to be BRDU positive, but it's not going to tell you, for example, that was a beta cell. right? Instead, EDU contains this alkyne group here, and we can perform a click chemistry. So the way the click chemistry works is that azids can interact with alkynes to form a covalent bond, and you can use there's a nice kit now out there, of course, where fluorophore is conjugated to an azid and can be directly attached to the incorporated EDU. So that's very straightforward and allows beautiful preservation of the cellular structure. Now, I know a lot of people like to use Ki-67 staining as well for proliferation. And actually, when we started our culture system, that was what we used as well. However, the more we thought about it, Ki-67 provides a snapshot of whether cells are in G0 or not, right? And the staining pattern of cells will look slightly different in different phases of the cell cycle. But it's really a binary snapshot of what's happening right now. EDU incorporation is a cumulative readout. So the longer you keep EDU in the culture system, the more cells that are dividing you'll capture, right? This turns out to be very bad for something like a cancer cell because you'll get 100% of the cells will be EDU positive. But in our case, for beta cells, it turned out we needed 6 days of incorporating EDU to see any signal. And to compare, this is a 5-IT is a compound we frequently use. And again, I'll get to that in a moment. But shown here are 2 nuclei from beta cells that are EDU positive and C-peptide positive as well. But you can see that one of the nuclei are is Ki-67 positive. The other, may or may not be. And so, you get different results. And I think that for our screening purposes, EDU has been enormously helpful for that because it raises our signal to noise and allows us to see over time compounds that actually do cause entry into this S phase of the cell cycle. Okay, so with that, I'm going to go over our assay flow that we use for beta cell proliferation screening. So first, as mentioned, we prepare the plates. So these you can store for about up to a month. Sometimes if we don't know we're getting islets and we're out of plates, we can prepare them the night before. That's okay. But what we do is we incubate 10 microliters per well. And again, this is all 3D 4-well plates, but this can be easily adapted to 96. We do that all the time. Now, we don't have a lot of experience with, say, like 48 or 24-well plates, but, this process is so straightforward. I don't see why it wouldn't work, but that would be maybe up to you to test. So 10 microliters per well of our conditioned media from the HTB9 cells, and we incubate at 37 degrees. We incubate overnight when possible, but that can be even 1 or 2 hours. And then we wash twice with PBS. The next day, presumably, we would receive our islet shipment, and we're going to seed those cells using a combi, which is from Thermo Fisher. It's an automated liquid dispenser into plates. I wouldn't really call it that automated. It's sort of semi-automated. And, in terms of lab equipment, it's not the most expensive thing in the world. So, you might consider, if you don't have something like this, it's pretty obtainable. So when we get the islets, we wash them, and then we use Accutase to dissociate them, and usually about 20 minutes at 37 degrees. And what's important here is that we do gentle dissociation. We do not try to get single cells here. And part of the reason for that is, just sort of empirical, that the beta cells like to have that cell connection still there. We don't want to have too many clumps, and we try to get the clumps down to about 10 cells per clump, for lack of a better term. But again, it's important to know the harsher that you dissociate the cells, that can be a real failure point in proper culture. So if you're going through this process and you see that they're not thriving, that could be a reason. We again then dispense 50 microliters per well of these cells. Over time, we determined that 10,000 cells per well in a 384 well plate is really optimal for us for beta cell proliferation. That's a lot of cells in the plates and in the well. And so for other readouts, you probably could get away with smaller numbers of cells, for example, like insulin secretion and so forth, or cell death. But because we're looking for a kind of rare event of beta cell proliferation, we need to really up the number of cells per well. And then the next step, so what we do here is actually, it's not 50, it's total of 50 microliters per well of cells. But what we're doing is we're always adding 40 microliters per well to 10 microliters that are in the plate. And so when we do aspiration, we always leave 10 microliters in the plate. If you let them dry out, that can be a real pain point as well. We leave the plates at room temperature for 30 minutes to let the cells settle. This actually has been something that was in the literature for screening purposes to reduce edge effects. And that the edge effect is the idea that on a 384 well plate, is there a picture of one? Oh, we'll get back to it. 384 well plate, because you have just so many wells that the edge around the entire perimeter can evaporate a little faster than the internal wells. And so your readouts can differ depending on the edge versus the internal. And so assay development, you have to be careful of that kind of edge effect. Leaving the plates at room temperature for 30 minutes kind of helps mitigate those effects. And then we incubate it at 37 degrees, of course. Now, then we do our screening. And here we have compound plates. Sometimes we would prepare them, but more often than not, we get them from our compound management group, or we already have them prepared and ready to go. Typically, we use 5 millimolar stock solution of these compounds in DMSO, and they exist at about 10 microliters per well. And the reason for that is when we pin transfer later the 100 nanoliters of compounds in each well, we're going to get about a 10 micromolar final concentration because it's a 500-fold dilution. So we changed the media to containing EDU as well. And what we use is it's called the ELX 405. It's from biotech. Again, it's in the protocol. It's a mid-range piece of equipment, but what it does is it allows you to automatically aspirate all the wells of a 96 or a 384 well plate. And then we add the EDU and then pin transfer the compounds. This is probably the most specialized step. Right now we use a SciBio Vario, which uses a steel pin-based transfer. Many screening facilities use also acoustic transfer, where actually you can take your plate of DMSO containing DMSO, where each compound is dissolved in DMSO, and use an acoustic wave to transfer nanoliters of compound to your well of containing cells. We have not really explored that very much, to be honest. So right now all of our steps are optimized to the plate-based and pin-based transfer. However, what you can do is you can buy even like yeast replicators, which are, the 96 or 384 pins, even plastic. And you can even use those manually in your lab to transfer compounds. Alternatively, if you're testing just a couple of compounds, you would just pipe them in. Okay, so after 6 days total treatment, and here we are changing the media and changing the compound every 2 days. We remove the medium, fix the cells, and again, always 10 microliters per well remaining in the cells, in the wells. And we fix with PFA, and we wash twice with PBS, and then we permeabilize with Triton X100. And then we perform the click reaction. This is all according to the manufacturer's protocol. It's pretty straightforward. This is a click chemistry-based reaction to attach the fluorophore to, remember, the alkyne of the EDU. And then we perform blocking and immunofluorescence using, again, primary antibody to, usually we do C-peptide, but I'll talk a little bit later about alternatives. And we use a secondary antibody to with a complementary color, of course, to the EDU, and nuclear mask to highlight the nuclei. We wash them, and eventually you end up with a plate full of 50 microliters per well of PBS, and we seal and store in a fridge until we image. For our imaging purposes, we use the Perkin-Elmer Opera Phoenix, but over the years, we've used many iterations of microscopes. So, I'm not going to say you must use this microscope at all, because I think any microscope will do. For us, because we're doing 3D 4-well based screening, an automated microscope was a must, though. And the Opera Phoenix contains Harmony, it's called a Harmony application that comes with the instrument, and that works real well for us. We used to use Cell Profiler sometimes, which is freeware that was actually developed by folks at the Broad as well. And but we just found that the canned software that comes with the Phoenix works real well. And the idea here is that we filter out the large nuclei, and I'll show you in a minute what those images look like. The fibroblasts, because it's 6 days, they're all going to incorporate EDU. So you get a lot of cells that look like they're green, but they're not beta cells. So we need to filter them out computationally, and we can do that by the size of the nucleus. So we keep the proper size nuclei that represent endocrine cells, and we can calculate fluorescence intensities and do those measurements. And here's an example of something that comes out of that process. This is an older image, but still looks great, I think. And it's only a 3-day here EDU incubation, but we've stained for EDU and insulin here. And what we found was that even in a control well, this is treated just with DMSO, we find very, very low but non-zero levels of beta cell proliferation. And circled here is an example of exactly what we're looking for, with red cytoplasm surrounding a green nucleus. These other cells are the ones I'm talking about that kind of overwhelm the culture. And fortunately, with our image analysis, we can filter those out because they would just introduce far too much noise otherwise into our system. In the past, we worked with Andy Stewart's lab also to overexpress, for example, CDK6 and cyclin D1, just to show, look, we can make these beta cells proliferate. This is, CDK6 is not what we want to use, but it's a detection positive control in our system, and allows us to say, hey, we can detect beta cell proliferation, and these cells are capable of responding. So if we're going to find a compound that does exactly this, we should be able to detect it. And I've told this story to many people, so you might have heard this already, but one of the compounds we tested was 5-iota tuberculin, shown here. It's an analog of adenosine, and it was originally developed as an adenosine kinase inhibitor. But we found that in human islets, and it was found and published even as an inducer of proliferation in mouse and pig and rat islets, but was never really discussed about humans. And what we found was that we saw very strong upregulation of these green nuclei, and even finding things like quadruplets of nuclei together, which to us felt like, wow, we're really watching. We believe we're seeing cells that divided, or at least nuclei that divided. Now, earlier I mentioned 6 days in EDU culture, and we did a time course to see, well, what is it going to take? Because for high throughput screening, it would be nice to do fewer days because there's logistical challenges of culturing them for 6 days. And since we're changing the media every 6 days and re-adding compounds, there's always the opportunity for error. And so, shortening it would have been better. But unfortunately, what we saw is that we 6-day culture for this as a positive control was really going to be required for us. You can see an increase here. And if you're testing one compound or a few compounds, then I would suggest 3 or 4 days should be fine for you. However, for us, this was not, for a high throughput screen, this was not a high enough dynamic range for us to computationally and statistically say with fair amount of certainty, wow, that's a hit or that's not a screening hit. So we went with 6 days. And we had a nice dose response. This is 5IT, where we've tested down to 30 nanomolar, but all the way up to 500 nanomolar. And we see this nice increase in response. These are just the controls on the edge of the plate containing 0 or 0.5 micromolar of 5IT. And when we run this on a plate, here's a sample plate, we see that the number of beta cells per well remains pretty constant. Again, as I mentioned, this is a nice case where we see about 55% of the cells are beta cells. We typically get on the order of, like I said, maybe 30 to 50. 30 would be not so good, but around 50. And when we look at the percent and the number of proliferating beta cells per well, we see we get less than 0.5% proliferating, and that increases. And this is where there is some donor variability, but this separation is not variable. This always happens, and some donor islets react much better, where we see, oh wow, 4% or more are proliferating. Sometimes we see 1, things like that. That's why for screening, we need it as a positive control on every plate. But we get this nice separation. I'll come back to what the ZPrime factor means in just a moment. Actually, I'm going to talk about it right now. So in high throughput screening, traditionally, over the last 20 years, this idea of a ZPrime factor has been raised. And what this tells you, it's not a Z-score. Z-score is for a compound, but a ZPrime factor is for the assay as a whole. And it's a measurement of this, basically the statistical power to measure small changes from a compound and to register them as a screening hit. There's a formula for it, there's a wiki page for it, things like that. It's really based on the idea that you might have a negative control and a positive control, and the distributions are going to be as such, and that you want to have a good separation here between 3 sigma in either direction. However, for us, we found this was only applicable. ZPrime is really thought of as applicable to a Gaussian distribution, sort of a bell curve shown here. However, with proliferation, this is a rare event. And really what we thought was actually a Poisson distribution is more accurate. And we've collaborated with a computational group at Broad that we work with all the time to develop a slightly different equation, and we're preparing a manuscript now. Hopefully that'll be out soon, but we can go in more detail offline afterwards. But the idea is that we can see the Poisson distribution and use that to model how well the compounds are responding, the cells are responding. We also built an online tool we call Grape for generalized rare event assay Poisson estimator. This will help you calculate, if you were to do, for example, a plate of compounds, to calculate the Z prime of your plate, and also to determine whether or not you have any screening hits in your system. Now, I mentioned earlier, we can co-stain with C-peptide and glucagon, for example, and this is a well that's treated with 5-IT. But I think you might be able to perceive right away that these cells are very closely opposed, and oftentimes it becomes difficult to tell which cytoplasm belongs to which nucleus, right? And that becomes a real challenge for us in our image analysis and our screening performance analysis, is to figure out, well, are they really beta cells dividing? Are they alpha cells? Are they other cells, that are other endocrine cells? To get around that, we tried to do staining with PDX1, thinking, well, it's nuclear, so EDU and PDX1 will be right on top of each other. That should be much easier to measure. However, we found something really interesting, which didn't work for screening, but has led to another project, which is that every time we found an EDU positive, C-peptide positive cell, it was PDX1 negative. So, and this happens every time we see EDU incorporated. This is not just based on compound treatment, but also even in the DMSO condition. Under the very rare state where we found an EDU positive cell in DMSO, it also lacked PDX1. We're doing another whole project around whether it's, which is the chicken and which is the egg, so to speak. Is PDX1 lost, or is it already gone? These cells do still have an endocrine identity. They have chromogranulin staining, but they've lost, or they don't express PDX1, MAFA, and KX6.1. And so we're charging through that now. In a lot of ways, it's probably not so surprising that a proliferating cell might lose some of its markers of maturity, but we think we can use this system now to really study what are the kinetics of that process, which is really cool. Here's an example I show here of, this is EDU cells, PDX1 is in yellow, and C-peptide is in red. Circled are the EDU positive cells, and I'll go back and forth, and you can see that every time they have a green cell, it's negative for PDX1. So this really, convinced us that this was happening. So that's it for proliferation. We've now performed actually a screen for 10,000 compounds, and we're following up on some hits, and that's really exciting. And we hope to get new targets. I know that many of us in the field have been trying to find some compounds to promote proliferation, and many times it all comes back to DYRK1A, which is a kinase that when inhibited can promote proliferation. Happy to say we found at least one compound that is not targeting DYRK1A, so I hope to have an update for you in the near future on its mechanism of action as well. I mentioned earlier that we can measure beta cell function in this as well, and so this culture system can be very useful for things like GSIS. Now, with an ELISA-based system, it's not going to be used, say, for high throughput screening, but you can use it to look at different compounds that you identify through other means, for example. And just shown here is the fact that 5-IT, for us, at least in acute phase, where we incubate for 2 hours with these compounds, has no effect on GSIS. And after 6 days of treatment, we found that it is more or less preserved the GSIS as well in these wells. Now, what we do here in this 2D culture system is we actually normalize by the beta cell number. So we take the supernatant to do an ELISA, and we stain the wells for C-peptide and use those values to help normalize the values we get in the ELISA. I'm not going to go into great depth at all about whole islet methods yet, because we're still it's really a work in progress. And so I think that it's, I'm going to close that. Something that I hope maybe we can give another webinar on in the near future. But the idea would be, what I've shown you here is entirely 2D culture system. Wouldn't it be better if we could screen intact islets? And we're working on those readouts right now. So as mentioned, good. The resources site of the HERN network page shown here can allow you to reach the resource browser. You can type this in, resourcebrowser.hernnetwork.org. And if you highlight document, you'll see that we've now deposited this islet cell culture protocol. This is also available on protocols.io, and we can share that link as well. And when you click on the protocol, it actually links out to the protocols.io. So there you go. It's a Word document. It goes through exactly what I've told you today, and hopefully it'll be really useful for you to test your own conditions. It doesn't have to be compounds. It can be peptides or growth conditions, metabolites, anything really. I think that this is something that that everybody should be doing, or when you can, when you need to, and but it's also something that everybody can do, right? We have new people come into the lab all the time. And I think as long as you're careful with your process and have good hands and just go slow, this seems to work well, really almost regardless of the islet sample we get. With that, I really want to thank my team and our computational team. Masha Alamova is a microscopy expert at the Broad. Deepika Walpita is now at Genelia Farm. She leads their cell culture core, but she's the one that really went back to the literature and found all these nice ECM methods. And so, she's done a great job to put this into place. And then since then, Toshi and Kumiko have been taking the charge in running full-scale screening. And so it's been a very exciting time. All right, with that, let's see, it's 2:40. So I'm really thankful for all of you for participating and attending, but we'll do the fun part now and have questions and answers.
That's great. Thank you so much, Bridget, for that talk and making this within reach. I felt like I could do it.
Yeah, and you can.
Yeah, thank you. So what we're going to do is we're going to take questions. And if you'd like to ask our speaker a question, go ahead and unmute yourself, and then I will see you in line and I'll call upon you. But what we'll do is we'll switch between the questions that are coming in live and the questions that people have asked in the chat. So while you're unmuting yourself, I'm going to go ahead and go to the questions. I'll go to a question that came in on the chat. There were a few of them, Bridget. One of the questions that came in, interesting when you were talking about your PDX negative, when you were seeing EDU and C-peptide positive. Actually, someone was making a comment that in your EDU positive that there would be a lot of C-peptide negative cells. And what do you do with those, and can you deplete those? Can you comment on that?
Yeah, absolutely. So we do deplete them computationally. So take the image and you can see, maybe I can go back and share the screen again. All of those cells and you can see that they're large nuclei. And, if we were to just analyze this by eye, it would be easy to see, but you can teach a computer how to do that also. And so, for example, here you see all these huge nuclei. These are not beta cells. They're not C-peptide positive, right? We call them fibroblasts just because it's easier. I'm sure there are many different cell types amongst these big behemoths out there. The small nuclei, though, are the endocrine cells. And so most of these that you see here are beta cells. We don't have a nuclear stain showing right here, but we do have alpha cells and other cell types in there too.
Great. Thank you very much, Bridget. That question came from Julia, by the way, last name not stated.
Great, thanks.
Another question that came in the chat, I don't see any questions yet, anyone unmuting themselves. This question is from Sasanka Ramanhan. And the question is, how long does it take for the cells to attach? So this came in at kind of the beginning of the presentation when you were talking about plating and then peeling away some of that. So could you comment on that? And also the question was, how long were they attached before you did your GSIS study?
Yeah, great. For our proliferation studies, we let them adhere overnight. So we plate the cells and then put them in the incubator, and then the next day change the media and add compounds. The GSIS, in the case of the acute 2-hour GSIS, that would have been overnight as well, but the 6 days is going to be then they're in place for 6 days. What we did find, though, is that sometimes especially for things like for readouts like survival, sometimes it's even good to give them one more day to adhere to see to allow those cells to, they settle down. I mean, it's not scientific really what I'm saying. They spread out, they seem to get more comfortable, right? So sometimes when we haven't found that to be the case in the proliferation, but in other readouts, sometimes that is true.
Great. Thank you. Thank you very much. Okay, I still don't see any questions coming in live, but I know the speaker likes to get live questions, so please
sure.
don't be shy. We'll go on to the next. I guess this wasn't a question so much as a comment from Stacy Weaver when you were talking about sending out these acoustic transfers. She said that the echo machines for acoustic transfers are amazing. So it sounds like that's someone that has some experience.
Absolutely. I can go in. Yeah, no, so it's a great advance for high throughput screening. And if you've ever seen, you should go look online for videos of it because it's a little disconcerting. The way it works is that you have your plate of compounds, and then you have your receiver plate that can either be a dry plate or a plate with your cells. And the machine flips it over upside down on top of the compound wells. And the first time I saw that, it was just like, but the capillary pressure within the wells keeps the media there so that it doesn't just all drip out, which is amazing. So I agree with Stacy there.
Yeah, that sounds pretty cool. Okay, a question has come in from Chung Yang Chen. And it was a question I was thinking about too, because you've been talking a lot about beta cells, but the question is, do compounds that stimulate beta cell proliferation also promote proliferation in other endocrine cells? Have you looked at that at all?
Yeah, and in fact, for our screen, that's always one of the counter screens we like to do. First, of course, we want to make sure they're not inducing proliferation of all cells. And that's where having those fibroblasts in the culture is helpful, because sometimes the compounds just make every cell proliferate, and so you can weed those out. Some compounds do cause pan-endocrine proliferation, and others don't, and so we're filtering to make sure we're focusing on the beta cell specific as much as possible.
Okay, great. Another question that came in from Damilola Adoya, I hope I've said that correctly. Very complimentary for your presentation, Dr. Wagner. The question is how you quantify cells. You mentioned something about 10,000 cells per well early in the presentation. Could you talk a little bit more maybe about how cell quantification happens? We
quantify them like you would in any cell culture system. We have a cell counter where, but I think, long time ago when I was even in the lab, we were using a hemocytometer. You can use those to measure your density, and then make sure that, you divide that by 50 microliters, because that's how many are going into the well. So your density ends up being, I think, in our case, might have been 250,000 per well. Anyway, it's in the Word document as well. But we quantify them the way you would any cell line. Yeah.
And that includes your methods for dissociating whole islets as well?
Yep, exactly.
Okay.
And we're going to get clumps. So, when I say 10,000, that's as close as we get. We estimate what we can.
Okay, great. Another question coming in from Dr. Parent, and she asks, have you tried using stem cell derived beta cells?
That's a great question.
I'm sure others are probably curious. Yeah.
We have. Every time we have tried them, they're quite proliferative. And so our background gets to be really high. We also saw that many times we don't do the differentiation ourselves, but we've collaborated with other groups to get batches from many different labs. So it's not just one lab, but even so, they don't secrete insulin very well, and in our hands, and they divide a lot, and that, I think those two would go hand in hand. So from a screening perspective, it's not, I don't know, we found it's not really ready, but it would be great to get to that cell source, because I think the number of compounds we can screen would be really much greater. I mentioned at the end that we had screened 10,000 compounds, which from an islet perspective is, it's amazing, right? But in terms of high throughput screening, it's awful, right? Like that's such a small number, right? We could do 10,000 compounds in a day with any other readout. So, the islet world, we have a big gap, and I think stem cell derived beta cells would be the right thing to do. So I agree, but yeah, we haven't had a good success with that.
Yeah. Okay. Wonderful. I don't see any more questions in the chat. I hope I didn't miss anyone in the live. Oh, it looks like a question just.
Yeah.
Oh, okay. Somebody agreeing with you. So
absolutely, yeah.
All right. Very good. Yeah, I hope I didn't miss anybody in the live participant section, but I didn't see any questions come in there. So yeah, I don't think there are any more questions. So again, Dr. Wagner, I'd like to thank you for presenting in the webinar. Thank you for sharing your information. And I'll remind people that Dr. Wagner has deposited this so you can get access to this protocol right away. And I'm sure she or members of her lab would be happy to answer questions or help you should anything come up.
100%. Just feel free to shoot me an email if you are trying to embark on this. Thanks.
Okay, thank you very much, and thank you everyone, and we'll look to see you next month for our next webinar.
Wonderful, thanks.
Okay, bye-bye.